So let me welcome all of you to this ADVAC webinar on behalf of ADVAC. I want to thank Paul-Henri and the speaker for accepting to chair kindly, and the speakers and the panelists to take off their time to help us with this webinar. Before I turn to Paul-Henri to briefly introduce him, I want to remind you the rules that unfortunately with the large number of participants, we cannot have everybody go live and ask questions. So rather, we ask all of the participants that are not discussants to kindly use the question and answer box at the bottom of the bottom bar to write your questions with your name, and we will ask the questions for you. And apologies if due to time we cannot have all the questions answered. I'm sure that with this timely webinar on the contextualization of the results of the early COVID trials, there's a lot of interest, as we have seen from the registrations, and a lot of questions. Please do not use the chat box if you want to ask questions. We cannot monitor the chat box, and the questions asked there will not be asked on your behalf. So without any more delay, I would like to turn to Paul-Henri. Actually, I said I would introduce Paul-Henri, but after being at the helm of ADVAC for 20 years, I think that Paul-Henri is well known to all of you. So I'm just going to ask him to take the chair. Paul-Henri, please.
Well, thank you, Philip. It's a pleasure to be with you again. And good morning, good afternoon, and good evening. I think that we will start right away. We don't have so much time. And I'm pleased to introduce the first speaker, who will be Peter Smith. Peter Smith, as you know, is Professor of Epidemiology at the London School of Hygiene and Tropical Medicine in London. He has been, in fact, ADVAC lecturer since about 20 years. And he is very much involved in the MRC, International Statistics and Epidemiology Group. He will give a lecture on early results from COVID-19 vaccine trials and implications for ongoing and future trials of other COVID-19 vaccines. So Peter, you can start. Thank you.
Thanks very much, Paul-Henri. I hope everybody can now see my screen. As Paul-Henri said, and welcome everybody. What I want to try and do is just give a summary of what I think we know about the vaccines that have reached and given some preliminary results of phase 3 trials, just to summarize those results as far as we know them. Then to discuss what the implications of those results are going to be with respect to the evaluation of the large number of other COVID-19 vaccines that have either are in the middle of phase 3 trials, or some of which have not yet started phase 3 trials, but which are expected to start in the coming months. And these early results from the first vaccines past the block, as it were, are going to have some considerable implications for evaluation of these later vaccines. So basically, in a phase 3 trial, as I think you all know, there are two principal questions that are to be addressed. First of all, and of primary importance really, is the vaccine safe? And secondly, and also obviously important, is the vaccine efficacious? And to address those questions, we generally required a double-blind randomized trial, usually with a placebo as the comparator group, although not necessarily. For example, the Oxford vaccine has used a meningitis vaccine as their control arm, a vaccine that will offer some benefit, but that will not be expected to affect the incidence of COVID-19. And principally, the results that the conclusions we want to come to initially with respect to these early phase 3 results is really do the short term, and these are very short term efficacy results, normally a follow-up of at most 2 or 3 months. Do these justify either licensure of the vaccine or more likely some sort of emergency use listing, which will allow widespread use of the vaccine before the vaccine is fully licensed? And the other question about these early results is really, will the characteristics of the vaccine that are being reported and the results from the phase 3 trials convince public health authorities to deploy the vaccine, and even more importantly, to convince potential recipients to take it up? Now, there's a number of important secondary endpoints which may or may not be addressed in the pivotal phase 3 vaccine trials. I think a very important one is, does the vaccine protect against severe disease and death? Because these are the endpoints of principal public health importance, and information on vaccines so far with respect to even severe disease, let alone death, is somewhat limited. We also need to know if there are subgroups in the population in which the vaccine is more or less efficacious or more or less safe. And the sort of groups that we're particularly interested in are the elderly, who we know are at increased risk of severe disease, substantially increased risk of severe disease, people with comorbidities, pregnant women who have been excluded from most of the vaccine trials, and those who are immunocompromised with HIV or other conditions. Very importantly is, how does protection change with time since vaccination? All we know at the moment is whether a vaccine protects for a month or two. We don't really know much beyond that. If there is some waning of protection, can we actually boost that protection by booster doses if necessary? Again, that's a question for the future. And with respect to safety, are the rare adverse effects of vaccination, which are too uncommon to have been detected in phase 3 trials? The phase 3 trials have been large, as we'll see, but if adverse effects are occurring in sort of 1 or 10,000, 1 or 20,000 individuals, they may not actually have been picked up in those trials, and that will only come from post-use surveillance. And then importantly also, are the late adverse effects of vaccine, most of the adverse effects of vaccines that we see and we know about of other vaccines occur within a month or two of the administration of the vaccine. And that's why FDA, for example, is insisting that on average there's at least two months of follow-up after the last dose before they're prepared to consider a vaccine with respect to potential emergency use listing authorization. But we know that there are some effects which may occur much later. And the one I think that has attracted most attention is the possibility of enhanced disease induced by vaccination, which we've seen previously for dengue vaccines that occur a few years after vaccination, when vaccine-induced immunity begins to wane, and is there the possibility of vaccine-induced enhanced disease occurring as vaccine-induced antibody levels decline with time? And we may not know the answer to that for 2 or 3 years. In terms of studies on MERS and SARS-1, there was some evidence in animal studies that in some circumstances such enhancement might happen. So far, with respect to at least my knowledge of all the animal work that's been done so far, there's no support that enhancement, there's no evidence of that enhancement in the animal studies, but it's a possibility that we can't exclude at this stage. We also don't know at this stage whether the vaccine protects against asymptomatic infection. the endpoint in most of the trials is in fact, the main endpoint in all of the trials is symptomatic disease. And we don't know whether the vaccine protects against asymptomatic infection. And that's going to be important in terms of the potential of the vaccine to induce herd immunity. If it protects against disease but not against infection, then that's a different sort of vaccine that also protects against infection. And then finally, and importantly, can a correlate of protection be identified? Because if we have a validated correlate of protection, then that may ease the licensure process for future vaccines, because as we've seen with other vaccines, we can then apply for licensure on the basis of the correlate rather than on a phase 3 efficacy trial. We're a little way away from this yet, but hopefully such correlates are going to be identified in the coming at least year. Some of these issues that I've listed above can be addressed in the phase 3 trials, but others essentially are going to be necessary to address in post-deployment studies, some which will be as part of the vaccine developers risk management plan. And Nick Andrews is going to talk about some of those studies in the next talk. Just to try and summarize what I think we know about the vaccines that are produced for generally interim results, or in some cases final results of their phase 3 trials. I have to qualify this by saying that most of the data in this slide is based on press releases. And so there's a certain amount of uncertainty, but we've seen just in the last day, further data published from Pfizer-BioNTech on the FDA website and in Lancet on the AstraZeneca, the Oxford vaccine. So we've got some more data, and I don't think anything that's in this slide is contradicted, at least in those two cases, by the data that's been published there. There's different platforms, 2 RNA platforms, and then 2 adenovirus platforms. We know least, I think, about the so-called Sputnik vaccine from the Gamaleya in Russia. Very little information on that, but they have published a press release giving these data. Most of the vaccines have an interval between, need 2 doses, both these and most of those in the pipeline, separated by 3 or 4 weeks. They've been quite large trials, some tens of thousands of individuals. They've mostly focused on adults, aged 18 and over, and the trial sites have varied. Some have been just in one country, US or 2, UK, Brazil for the Oxford vaccine, although other sites are planned, and one in Russia. And then the Pfizer trial was a multi-country trial. They've generally randomized a vaccine placebo or vaccine control 1-to-1. An exception is the Russian vaccine, that's 3 vaccine to 1 placebo. And in terms of their primary endpoint, they've generally defined this to be either a week or 2 weeks after the second vaccine dose. And based on those criteria, the efficacies that have been published, shown in red there, with the numbers of cases in the vaccine and the control group shown in brackets. So highly efficacious vaccine from Pfizer-BioNTech, 95% efficacy, only 8 cases of symptomatic disease in the vaccine group compared to 162 in the control arm. Not dissimilar findings from Moderna, 94% efficacy, somewhat lower efficacy in the Oxford vaccine, 70%, still highly significant. And a complication in the interpretation of that trial is that by mistake, some individuals got a half dose of the vaccine as their first dose rather than the full dose. And curiously, they report that in those that got the half dose, the efficacy was 90%, whereas those who got the planned regimen, it was 62%. It's going to be interesting to see what the regulators make of that difference. And then the Russian vaccine, again, 91% efficacy. The most important endpoint in many public health respects is severe COVID-19, and we know less about that. In the Pfizer, it's 1 versus 9. Moderna is more impressive, 0 versus 30. In the press release for the Oxford vaccine, it's 0 in the vaccinated group and an unknown number. We now know that number, and it's depending how you count it, it's 1 or 2 in the control arm. So that was predominantly a young population that vaccine was tried in. So we really don't have much data with respect to severe disease, although there were 10 cases of hospitalized disease, not classified as severe, and all of those were in the control arm. And we don't know anything about the Russian vaccine with respect to severe disease. The planned follow-up time is between 6 months and 2 years. Whether they're going to be able to preserve a placebo group or a control group for that time is problematic, as we'll see, and I'll discuss in a moment. With respect to the availability of this vaccine, these are the sort of estimates of the production capacity that the companies have. In this year, fairly limited, 50 million of Pfizer, 20 million of Moderna, and during next year, of the order of 1.3 billion for Pfizer, half to 1 billion for Moderna, more for AstraZeneca, Oxford, 3 billion, and an estimate of 1 billion for the. These are large numbers, but as we'll see shortly, they're still not enough to cover the world, as it were. They have very different characteristics, these vaccines, with respect to their, if you like, their usability. The vaccine that's most advanced, the Pfizer vaccine, has to be stored currently at minus 70 degrees, so a real challenge in getting that out to populations, and it's only okay at fridge temperature for up to 5 days. Moderna similarly has to be kept at minus 20, a bit better, but still, and at quite longer shelf life in the refrigerator, whereas the AstraZeneca and the Russian vaccine have much longer half-lives in the refrigerator. And insofar as one can actually work out the costs, somewhat variable costs, the Oxford vaccine being cheaper in terms of current estimates than either the Pfizer, Moderna, or Gamaleya, though as we've seen previously, costs do tend to change. Those are the vaccines that have actually given interim results. These are vaccines that are in phase 3 trials. You can see in the last column just when those phase 3 started. Quite a lot of different vaccines here. Large vaccine trial by Janssen, another vaccine trial by for AstraZeneca Oxford, generally including more older adults than was included in the stuff that they've published so far, and then other trials here, both in China, India, and in fact, this morning, there was a report from Sinopharm in UAE that their vaccine is showing in an interim analysis 86% efficacy, but no further data on that. Note that virtually all of these vaccines, with the exception of one trial of the Janssen vaccine and Casino vaccine, Chinese vaccine, are 2-dose vaccines. So obviously we would ideally like a 1-dose vaccine, and if these work, that would give them quite a big advantage. But at the moment, most of the vaccines under development are 2 doses. I wrote this slide for another lecture I gave, and I said that participation in a vaccine trial should be driven by altruism. Vaccine trials are not conducted for the benefit of those in the trial. They're conducted for sort of those who will come afterwards and benefit from the results of the trial. So that in trials, participants often benefit from somewhat better care, and they obviously have a chance of receiving a vaccine which may or may not be safe or efficacious. And I postulated that participants should not be disadvantaged compared to those in the same community who are not included in the trial, but nor should they be unduly advantaged. And that relates really to the speed with which trial participants may have access to a vaccine once it is shown to be efficacious. I had to modify this slide slightly because it's quite clear from reports that not everybody is entering these trials based purely on altruism, and some people are actually driven to enter these trials because they want early access to a vaccine that might be efficacious, and that, as well as altruism, is a motivating force for entry into some of the trials. And I think these issues relate to considerations as to really how long it's going to be possible to preserve a placebo group in ongoing trials, or a no, as it were, a no vaccine group in ongoing trials. We need that group because if we're going to assess whether efficacy declines, the best way of doing that is in the context of the randomized controlled trials that are already set up. And it's an important issue for the public health assessment of these vaccines in the long term. We've got a vaccine which is highly efficacious, but after 6 or 1 year, it actually has very low efficacy, then that's very different from a vaccine that continues to have higher efficacy. So we really need to preserve the placebo group for as long as is possible in these trials in order to make those assessments. And there's an even bigger problem for vaccines which have not yet been in trials for long enough to see even short-term efficacy results, or the vaccines that are slightly behind in terms of development and have not yet entered phase 3 trials. If they can't do trials in which there's going to be a placebo group, then evaluation of efficacy and safety is going to be potentially compromised if they can't actually maintain a placebo group in those trials. And this has been of sufficient concern that the International Coalition of Medicines Regulatory Authorities, which is a sort of coalition of different regulatory authorities, put out a statement at the end of last month, and to the effect that unless maintaining participants in their randomized treatment group, vaccine or control, after a vaccine is approved, is clearly infeasible, we recommend that clinical trials should proceed as initially planned, with a follow-up of at least 1 year or more from completion of the dosing schedule, so that they're recommending there that there should be at least 1 year follow-up, preserving a placebo group if that is at all feasible. They do recognize that the feasibility of that is going to be challenging for some vaccines, but there are circumstances in which it might be possible to do that. And it's going to be very much individual vaccine, individual situation specific, which they recognize. And that's already began to sort of have repercussions, the publication of early efficacy results. This is just a clip from the Wall Street Journal that already people who believe they got a vaccine during a trial say they want to leave it and get the real shots once they're approved. And Johnson & Johnson, associated with the Janssen vaccine, doing these very large trials of their vaccine, sought guidance on the matter from the Committee of Independent FDA Experts on Vaccines, and they expressed concern that as it were, the approval for emergency use of the first vaccine would compromise other companies' ability to recruit volunteers for their own studies and make it harder to keep volunteers if they're eligible for these first licensed or authorized vaccines. Now, this is not a new problem, that how do you do trials with a placebo if you've already got efficacious vaccines? And there was a WHO report on this published in 2014 of considering situations in which use of a placebo in vaccine trials may be justifiable when there's already an efficacious vaccine. We've seen that particularly with respect to some vaccines, for example, rotavirus vaccines. One criteria is where a country wishes to develop a locally affordable vaccine, and that was the basis for the trial of the Rotavac vaccine that was trialed in a placebo-controlled trial in India after there were already licensed vaccines for rotavirus in other parts of the world, where you want to evaluate the local and safety and efficacy of an existing vaccine. And so placebo-controlled trials of both Rotarix and Rotatec were done in Africa, even though there were efficacy results from outside of Africa because of uncertainty about just how efficacious they would be. testing a new vaccine when an existing vaccine is not yet in local use. And that was the case of the Rotacil vaccine in Niger, in which a placebo-controlled trial was done just before they introduced other rotavirus vaccines in Niger. And then there have been vaccines trials that have done determine using essentially a placebo or no vaccination group, determine the local burden of disease when there's uncertainty about what impact the vaccine may have, so-called vaccine probe studies. So what are the options for further evaluation of SARS-2 vaccines? One is the continued blinded follow-up of participants in existing trials until they become eligible for vaccination in the national program and wish to receive a vaccine nationally. So generally, most countries are going to prioritize either healthcare workers or the elderly for initial vaccination, given the shortage of vaccines in the coming months. And so that those individuals may choose to leave an existing trial, have their vaccine placebo code broken, and receive the vaccine as part of the national program. Whether one can keep the rest of the individuals in the trial until they become eligible under the national program remains to be seen. That depends to some extent, I think, on what commitment was given by the sponsor at the start of the trial. I think, for example, in the Pfizer trial, participants were told as soon as we've got a licensed vaccine, then all of those in the placebo group would actually get be able to get the vaccine, irrespective, presumably, of what age they are and their, at that time, national eligibility. Of course, that does depend upon the when the vaccine actually does get a full license, if that's how that is interpreted. The second possibility is to conduct placebo-controlled trials, including only individuals who are not yet eligible for vaccination in the national program. So that in many populations, we know that throughout 2021, there's likely to be insufficient vaccine supply to vaccinate more than about 20% of the population. And those will generally be those individuals who are at most risk of severe disease. So that leaves the rest of the population who could ethically be included within a trial. There are problems with that, of course, that if you've taken out the high-risk individuals, you will only be able to conduct the trial in, let's say, younger individuals, and it will be necessary to bridge the efficacy results for those vaccines from the trial population to population groups, let's say elderly who would not be in the trial, and that may be challenging given we don't have a validated correlate of protection yet, and it may be necessary to do safety studies, which may be uncontrolled, in those population groups who are not going to be eligible for those trials. Another possibility would be to do head-to-head efficacy studies of a new vaccine against an existing vaccine. Seems to me that's unlikely to be feasible because of the very large trial size that would be required to, as it were, show non-inferiority with respect to those vaccines that we already have, which appear to be really quite high efficacy. And it may not be easy to recruit volunteers into such a study if the offer is this new vaccine, whether we don't know whether it works against this existing vaccine, which we think has very high efficacy. So those, although in theory one could do that, I just wonder if that's going to be possible. Of course, what we'd really like to be able to do is evaluate a new vaccine based on an immunological correlate of protection. First, the big challenge is to identify such a correlate, and then also be reasonably sure that it can be used across different vaccines, including those using different antigen delivering platforms. And that's not immediately clear that will be straightforward. And then another possibility, which is still being discussed, is the compare the efficacy of new and existing vaccines in human challenge studies. Obviously, you can only do those studies in individuals who are at very low risk of serious disease from challenge, and there's the difficulty of bridging from efficacy in this group to those who are at higher risk of severe disease, who you're most interested in having a vaccine which is efficacious, but that is an option that may or may not be explored. And just finally, just to sort of summarize where we are with respect to vaccines, these are, this was taken from Nature, published last week, just showing what the availability of vaccines is likely to be, and and what the pre-orders for vaccines, mostly by high-income countries, have been. So on the left, it just shows the vaccines at the top where interim results have been announced, and in the red or brown, it shows the numbers of doses that have already been pre-ordered, with the possibility of expanding those orders. So AstraZeneca, over 3 billion have already orders in place. And the blue shows what the production capacity is estimated to be in 2021. So you can see for most of those vaccines, the planned production for 2021 is in many senses already spoken for. There are other vaccines in the pipeline, and hopefully those are going to come on board as we see at the bottom, and there'll be other vaccines there too. And on the left, it just shows the countries who have been buying up these vaccines. Top of the list is Canada, who they've already pre-ordered enough vaccine to give everybody in Canada 9 doses of vaccine. So hopefully there's going to be some left over, and they can allocate that to low-income countries or whatever. And similarly, with respect to the US, United Kingdom, and Australia, and so on, they've been brought up a lot of vaccine before they know the actual efficacy results. But if you like, as they produce efficacy results, much of the supply of those vaccines is going to be spoken for. And down at the bottom is the COVAX facility, which is aiming to make vaccines available for predominantly low and middle income countries. They've secured so far 700 million vaccine doses, and they want to secure by the end of 2021 2 billion doses, with the aim of providing coverage to around 20% of the population of countries participating in COVAX, which is most countries now, by the end of 21. So that by the end, although some of the high-income countries are going to be pretty well vaccinated, probably by the early part of next year, the low and middle-income countries are probably going to have a coverage which is probably not greater than 20% if COVAX works by the end of next year. So I'll stop at that point. Thank you.
Thank you, Peter. That was a remarkable lecture. I think that we will move immediately to the second lecture to be given by Nick Andrews. Nick, as you know, was also involved in ADVAC for many years as lecturer. He is now deputy head of the statistical unit at Public Health England. And his title is Approaches to Study Vaccine Impact Post-Licensure. That means both vaccine effectiveness and safety. Thank you, Nick. You can share your screen.
Thank you very much, Paul-Henri. And I seem to have started on I'll just go to the first slide. Can you see my slides?
Yes.
Okay, sorry, let's go to the intro slide. Okay, thank you, and for the invitation. So I'm just going to go through the different approaches for post-licensure surveillance. I should note at this point that many countries and organizations have been working on plans and protocols and giving information around how to do this. And I've listed a number of them here that I've either spoken to or are aware of. And of course, the manufacturers themselves will have their phase 4 plans, their risk management plans, as Peter mentioned, and haven't kind of gone into thought about that at all, but I know they'll also have those requirements when they do have licensure or emergency use licensure. Peter actually, in his slide where he was talking about the secondary endpoints, did actually cover a lot of the questions that actually post-marketing surveillance would need to address, because the trials often can't. You know, you're talking about looking in other populations, the long-term effectiveness and the safety for the rare events. And the areas that you kind of overall would be thinking about with post-marketing surveillance not only cover this effectiveness, impact and safety that I'll focus on, but clearly continuing to monitor epidemiology of the disease, serological surveillance as well to understand how that's impacted. And clearly we need to understand what the coverage is in the population and the uptake of the vaccines, and also how this all feeds into modeling of future interventions and non-pharmaceutical interventions based on how successful the vaccination campaigns and the effects of the vaccine are. One of the things that's been absolutely key before we've kind of reached the point which we are now at having to do this assessment is to make sure we can get hold of the immunization data. And I would encourage everyone who's in this area to understand this, because without actually knowing how you're going to obtain immunization data at an individual level from your population, doing the studies would be, you won't get anywhere. And so this could be through a particular immunization registry, perhaps one specifically set up for COVID. Obviously, there'll be current systems in place for other vaccines. It might be through vaccination cards, potentially, in some countries, and even in the UK here, we are issuing a card. But I wouldn't imagine that would be the main source we'd use for many of the data sources. Or it even could be questionnaires when patients actually present with their illness being filled in at that time, and also getting back into health records. It's important that however the data are originally collected, if they can get it back into health records, then that's also ultimately an ideal source, because it's already there linked in with some of the outcomes you might want to look at. And ideally, this should be as timely as possible. So we've spent a lot of work actually in the last few months ensuring that the data are going to be available quickly. So one is knowing we're going to actually even be able to get hold of that information. The next is to consider actually what are the data sources and then the methods that you might then use to do this post-licensure surveillance. So for you need to know who the populations being targeted are and how to identify the disease or the safety outcomes in an unbiased way, and also consider what the comparator group might be, and their exposure group and any confounding variables that you'd want to take into account. And it really, based on those data sources and where you will find your cases and safety outcomes, that tends to dictate the best designs to use. And there may be a number of different sources that would require different designs. So I'll start on the effectiveness. First of all, actually the number of different outcomes we would be interested with the vaccine effectiveness. There's the symptomatic disease, which is what the trials have tended to focus on. And then there's the infection, where you're potentially picking up asymptomatic as well, which you might identify through regular PCR swabbing or looking for seroconversions based on blood samples. Then there's the sort of severity side, the more severe disease through hospitalization or mortality. And then there's also the question of transmission. Does the vaccine actually affect transmission, which is very important for the population effects of the vaccine. And so considering those different outcomes and the different questions and data sources will dictate the design. Now, there are maybe quite a number of different data sources that you would look to use. There's a lot of laboratory confirmation going on with COVID, and those cases being collated into national databases. And so that's clearly going to be a source that can be used. Then there might be general practice records where ideally this lab confirmed data is also getting back into these records. And then that's part of a cohort. And in fact, some general practices might have sentinel surveillance where they do specific swabbing, where additional information is obtained on the cases being swabbed and additional virology might be being done. There's also hospital records and reporting from hospitals, whether it's severe acute respiratory infections or intensive care unit reporting of cases. And then the last one is there might be actually defined cohorts that you can use or set up to monitor effectiveness. There may already, an ideal scenario is where you've already got some cohorts set up, such as in healthcare workers or maybe care home residents and staff. I'll mention a couple of those examples in a minute. And doing a detailed follow-up of a number of cases and potentially the families of cases that do occur can actually help understand better about the infectivity according to whether someone's been vaccinated or not, as well as the onward transmission risk if you can do sort of contact studies. And finally, there's outbreak investigations. If a large number of cases do occur in a closed setting, it might be possible to do to have vaccination as part of one of the variables assessed during an outbreak. So lots of different sources of data. In terms of the methods you then apply, that's going to depend on all those things. I will mention briefly randomized studies post-licensure. There is, the question, and certainly this is being considered potentially within the clinical practice research data link in the UK, about whether it's possible, if there's more than one vaccine, to actually have a randomization at say individual or cluster level within during the rollout. I'm going to focus more on the observational methods within the types of data sets I've just described, and these would include test negative case control methods, which we've often used for influenza, whether those who are testing negative are our controls and those who are PCR test positive are the cases, and you can use that as a case control approach. Or you could have alternative population or hospital controls. If you can identify suitable controls, can be difficult to know if they are suitable. And then with the cohorts that are being followed up, whether it's the GP cohorts or the specific cohorts, such as the healthcare worker or care home cohorts that you're doing regular follow-up in, that'll be a cohort person time type design. And then as a potential more rapid analysis, you can also use the screening method, where you're ascertaining vaccination status in your cases, and you can compare that to matched population coverage. And finally, there's the ecological approaches, which are more vaccine impact approaches, where you're looking at a before and after introduction to a particular population, looking at the incidence. We'll also want to do quite a lot of potential stratification and confounding adjustment. Stratification, if the numbers allow, into subgroups that you're interested in with specific health conditions, by ethnicity, age groups, and importantly, considering those with and without evidence of past infection, which if you can link to at least if you've got a large database of PCR confirmed cases, you should be able to identify through linkage whether they've previously been tested positive. More difficult to know in terms of blood sampling, because that would have happened to a much lesser extent, I would imagine. And the sort of confounders you'd have to consider at least would be sort of age, time period, region, health conditions, and healthcare usage, and potentially influenza vaccination as well. Particularly if you're using the test negative case control design, then there might be a relationship between those getting the flu and the COVID vaccine. And if you're using influenza cases as your controls, that needs to be adjustment for influenza vaccination status would be important. Just a quick examples of the planned studies in the UK. I'm sure others are doing having similar plans. We've actually added a vaccine question to the test request form for anyone who has symptoms of COVID and is requesting a test, or for any other reason they're requesting a test. We're now asking if they've had vaccination and finding out about doses and timing, and that would enable us that will enable us to do a rapid test negative case control assessment. And And also, if we're capturing the coverage data, we can do that as well, a rapid screening method based on the population uptake as we monitor that in real time. And that can be done based on looking at the vaccination status of our hospital and community cases. And then there's the cohorts. So we're using, for example, the Royal College of General Practitioners cohort, where we can do, where they do capture all of the testing data gets into that. So we can do a cohort analysis, and within that test negative case control, because the negative tests are also getting reported into that. And then the specific studies within healthcare workers, for example, in the UK, they'd already set up nearly a study that they're recruiting up to 100,000 healthcare workers with regular PCR testing every 2 weeks and blood testing every 3 or 4 weeks, I think. And that enables them to pick up asymptomatic infections, which will be very important for doing the infection vaccine effectiveness assessments. And so we can link all these individuals to their vaccination status, and also collect it on questionnaires. So that gives us vaccine effectiveness against infection, as well as the asymptomatic. And finally, one of the other studies is a transmission study where we will recruit vaccinated and unvaccinated cases and then follow up their households to see whether there's PCR positivity and within the household, and also doing antibody testing to identify seroconversions. So you can look at effectiveness against transmission, which are all the things which the trials are currently not giving us information on. In terms of surveillance, if we continue our current surveillance, then that does enable us to identify whether the change in disease rates in the vaccinated cohorts differs to the unvaccinated, essentially. And that will be evidence of population effects, more difficult to interpret though, and whether or not herd effects are expected, and maybe after a delay you see herd effects in other groups. But because the incidence is rapidly changing over time due to other interventions, it does become more difficult to interpret those data. In terms of methodological issues, there's questions about how appropriate test negative controls are. You want the assays that you're using, or the methods, the PCR, to be reasonable sensitivity and good specificity. That might not always be the case. Sample size, actually the fact that the VEs are coming out at 90% plus for some does help to need smaller numbers potentially, which is good news. One of the questions is, could there be behavioural modification in those vaccinated? That would be quite difficult to deal with and measure. And understanding who are the specific populations being targeted. For example, if the vaccinations initially rolled out amongst patients who are actually in hospital or visiting hospital, that might be quite difficult to deal with. So I'll quickly move on to safety. So common and less serious events are in the trials, but we're interested here mostly in the serious adverse events. And we go through this signal detection, strengthening, and evaluation is how I like to think of it. I won't go through, this is a slide I show in the ADVAC course actually when I talk about the self-controlled case series method, and it just goes through how the trials I identify mainly reactogenicity, and it is through your pharmacovigilance and other methods you rapidly identify signals, which then you go through a strengthening assessment and then careful epidemiological studies to actually validate and confirm associations. The detection step of it, clearly that happens through your vaccine pharmacovigilance, and countries have this in place, this passive surveillance. What in the UK we have called our yellow card system. But on top of that, active surveillance for detection is being incorporated either within databases, such as the clinical practice research database, a GP database, where you can compare your observed rates post-vaccination to background rates, or rates in specific post-vaccination windows, and a sort of an acute period maybe compared to a period a bit later. In other countries, cohort event monitoring, where you actively follow up a cohort, can be used to identify signals as well. by through carefully monitoring admissions to a hospital, for example. And because this testing is done frequently, you tend to have to use methods to adjust for repeat tests, such as the sequential probability ratio tests. And it's usually done, this active side, on a predefined set of events rather than with a passive, which is capturing everything. Many signals, I should say, will arise unexpectedly from other sources. So despite what you set up, be ready to find out from others and keep an eye on what else is being thrown up, because it's likely it may come from an unexpected source, such as the narcolepsy and the pandemrix vaccine. There's rapid assessment is then required when signals do come up around causality assessment, and there's tools for that from the WHO. Is the signal seen in other countries? Can you do an ecological study looking at rates before to after vaccination? Compare rates if there's more than one vaccine between the vaccine manufacturers to see if there's similar in the groups, and just looking at the timing of the events. And maybe if it's not an event you've already looked at, a rapid observed versus expected analysis within a data source, and getting input from specialists is essential. So this is the sort of what hopefully you can be done rapidly when something happens. But ultimately, often, if you really want to be sure about whether there's a true association, then you would use data sources to do well-designed studies, either, for example, within GP data, if it's data cases tend to present at GPs, using cohorts, case control, or self-controlled case series methods, or if more serious events that mainly present at hospitals, then having linked that hospital data linked to immunization data can enable you to use those data. Again, often the best method being the self-controlled case series or case coverage methods. And for some events where they're not clearly captured in those sources, it might be that you need to use specialist registers, such as we did with narcolepsy. We went to sleep centers. And doing code validation, where you are using electronic registers, is often important to address that. I don't want to spend too much more time, so I'll allow us time for questions. In the ADVAC course, I talk about self-controlled case series. It's great for rare events and just requires an unbiased set of cases. And so that's a method I suspect will be used in studies. But there will be methodological issues, particularly around, again, who's actually being targeted with the vaccination, and whether they're the frailer population. And the pandemic itself might have changed some of the baselines for our observed expected analyses, so that could be a challenge. And if different vaccines are given to different populations, comparing across vaccines would be difficult. And ideally, you want separate data sources to identify and test hypotheses. You might not want to use all your data to scan for identifying signals. So I'll finish there and just thank those people on this slide for conversations all around this subject. Thank you.
Thank you very much, Nick. We can now immediately start the panel discussion. That means that we will call members of the panel, and we will open the discussion to panel members first, before opening to questions from everyone joining the webinar. So first, I would like to ask Stanley, who is with us still for some time today, I think, whether he has comments or questions for the two lectures.
Yes, well, so after two such excellent talks, it's difficult to give a prepared talk or criticism, but let me give you, if I may excuse myself, with some random thoughts about this situation. And they're not in any particular order, but so first, the AstraZeneca trial, which was not well done in many respects, does provide us with a significant opportunity to derive correlates of protection, because, as you know, the in the study, there was unforeseen regimen of half dose prime and then full dose for the second dose. And the efficacies were quite different between the full dose and the half dose. Now, I suspect that has something to do with priming. That is that the full dose may have given too much of a response to the vector and may have decreased the value of the second dose. But be that as it may, whatever the reason, investigation of the immune responses to those two regimens would be quite useful to try to determine what the correlates of protection are. The second related point is that we do have data from the Russian adenovirus vaccine where they, I would say rather intelligently, gave two different adenovirus vectors in the first and second doses, and allegedly had much better responses and efficacy. That possibility does deserve exploration, whether two different vectors might be preferable to single vector regimens. Another point is, of course, there are many other vaccines that are going to be tested, one of which actually is a company that I have been advising called Contextualization, has a live attenuated strain of SARS-2. And it will be indeed very interesting to see what responses, both systemic and local, are produced, will be produced by this vaccine. Now, an important point which has been discussed by the two previous speakers is how to test new vaccines. As I think Peter mentioned, the possibility of challenge studies is, in my view, attractive because while it would not give us information on a large scale, it would tell us whether a new vaccine provides better protection to the mucosa than prior vaccines. And that, of course, is an important point, which we do not yet know for the vaccines that have already reported efficacy data. But if those vaccines do indeed prevent mucosal infection, any new vaccine, in my view, should do the same thing, or else should not be used. So that is something that really needs exploration. Now, Peter mentioned correlates of protection, and as one who has perhaps worked a lot on that idea, that to me is a critical issue, both for short-term and also long-term, because it would also allow us to determine what the duration of efficacy is likely to be by following the correlates with time. Another point which I've begun to think about is the long-term future. Now, if we are to control this virus, now, of course, the virus might go away by itself because of vaccination and infection, but we don't know that yet. And it may very well be that it becomes a constant factor. That is, infections occurring elsewhere in the world perhaps might be transferred to countries that are using vaccines, but not everybody in the country will be vaccinated. Which brings up a point regarding children. That is that although children are the least likely to develop symptomatic disease, some of them do, and some of them develop serious disease, as you know, the Kawasaki-like syndrome, etc. Well, if we were able to vaccinate children, and being an American, I would recommend that that be mandated. If we were able to do that, we might be able to establish an immune population that would grow and also give some protection to adults who may not be vaccinated. This is a strategy that has been used with rubella, because although rubella can cause complications in children or adults, by and large, it is not a severe disease in children, and yet we recommend vaccination in order to protect the adults, obviously, particularly pregnant women. So by analogy to that, it seems to me that vaccination of children in the long term might be a very useful idea. We do need to know a lot more about the mucosal responses, and that could be determined now, really, by challenge studies as well as by observation of mucosal antibodies and other means. There are many questions for which we do not yet have answers. And the data from the efficacy trials, I hope will be more, that there will be a lot more data from those trials. I hope that that samples are being taken which will allow analysis of long-term immune responses, and as I've mentioned, the correlates of protection. And the last point I would make is that, yes, we don't know the duration of protection, and it is, in my view, unlikely to be forever. So we do need to determine what can be done with vaccine boosting immunity by vaccination. And perhaps we will need a combined influenza SARS-2 vaccine that would be given annually, or maybe not, maybe every 2 years, or who knows. But I do think that we should start thinking about the future of this epidemic. And I will stop here.
Okay, thank you, Stanley. So, Peter or Nick, do you have any comments to make after Stanley?
Valerie, if you allow me, before leaving it to Peter and Nick to answer, I wanted to bring your attention to a comment and question made by David Goldblatt, which relates very much to the initial point raised by Stanley. And so if you allow me, I will read the comment because it pertains to the AstraZeneca vaccine. And David says that the vaccine shows no efficacy against asymptomatic disease, that they did regular are on the subset as per the Lancet paper, but this does not equate to no protection against carrier status, as lower viral titers may lead to less transmission. But the most important point perhaps that David is giving and is actually a question for Peter, and he's saying the AstraZeneca's immunogenicity shows very similar immunogenicity data for the low and the high dose schedule in both young and old. But indeed, the efficacy appears quite different if primed with the low dose. And the fact that the immunogenicity appears very similar complicates the discussion about the correlates of protection, as there seems to be a mismatch. And David is asking specifically Peter what he thinks about what's going on, and is clarifying that the immunogenicity in his question is both T cells, Ig, and neutralization titers, and that there is no correlation between Ig to the adenovirus itself and the response to the second dose. And that again, the immunogenicity data in the second Lancet paper is both with young and elderly from the Oxford team. Over.
Peter?
Yeah, with respect to the Oxford data, I haven't looked at their full paper yet, but I have looked at some of those results, and I think they're going to be very difficult to interpret definitively. And I personally have some reservations about putting too much weight on this supposed difference in efficacy according to the size of the initial dose. You look at, first of all, that was unplanned. And so this was not a planned analysis. And their a priori hypothesis was they put the whole lot together, and they agreed that with the regulators. And presumably if they found very low efficacy in the sort of half dose, full dose, and better efficacy in the other, they would have argued for sort of leaving that group out altogether in terms of evaluating the vaccine. So if you actually look at that comparison between those two groups, it's, according to my calculations at least, on the borderline of statistical significance. And given that this was one of many unplanned analyses, I am cautious, particularly as there's just, as far as I can see, there's no really good immunological data backing up that difference at the moment. Now, I think they argue that they do show a difference with respect to asymptomatic infection. And basically in the sort of half dose, full dose group, they show some effect on asymptomatic infections, and they don't show that in effect in the high and the normal dose group. Again, those numbers are very small, and it's far from being a significant difference between those two groups. And if you look at the data overall, I think you have to, at least I would come to an equivocal position as to really there is very convincing evidence whether this vaccine does have an effect on asymptomatic disease. So I'm not an immunologist, and I'm sure the immunologists are going to have a lot of time sort of discussing what the mechanism for this could be. But looking at these data statistically, and of course, everything that is not significant, I mean, it's not true. But I think one has to be very cautious interpreting these data at the moment, because they weren't expected. I sort of take, well, I'm going to be very interested to see what the regulators make of this, because I personally would be very surprised if the regulators would say there's enough data in this trial to say, well, the sort of half dose, full dose group is the best one to go with. The numbers just, to my mind, don't bear that up. And I would be surprised if the regulators come to a different conclusion, but we'll see. And there is a larger trial. And the other problem with that group, that trial, was that was confined mostly to younger people under the age of 55, I think. And there is a larger trial with the vaccine going on in the US, AstraZeneca, including a higher proportion of more elderly people. And that's going to be, I think, important in terms of overall interpreting those results. So I think the AstraZeneca trial is a much more complex trial to interpret than the Pfizer-BioNTech trial, and I think is going to be much more problematic for the regulators as well. Over.
I just would mention that the Russian trial, if you believe what they are saying, gave high efficacy with 2 different adenovirus vectors in sequence.
Nick, do you have any comment on this point?
Not on this point, no. Sorry.
Oh, well, maybe other point made by Stanley.
I mean, the main thing from the post-licensure point of view, we'll be doing our best to collate this information, and on the correlates, you know, we are looking at whether we can identify correlates, certainly from the disease itself. From the vaccine side, more tricky, but that's ongoing work.
You know, just one other point I meant to mention, and that is that we will need a test for infection in those who are vaccinated. The most obvious would be to look for responses to the nucleoprotein, which is absent in most, but not all vaccines. But that will be a very important methodological point for the future.
Peter, any comment on general comment in relation to what Stanley told us?
No, I think I actually agree with everything that Stanley said.
Very good. Okay, I think that now I would ask Peter Niels to come with some regulatory vision on this.
Well, I fully agree with Peter that from a regulatory viewpoint, a by chance finding of an accidental issue will not lead to an acceptance on a regulatory part. Of course, I didn't see all the data because they are still secret, so I don't know what really happened, how big the numbers are, etc. But if something is not planned in the protocol, regulators will look at it very curious and see what happens. But I would really doubt that they would go for a positive opinion on the half dose plus a full dose based on what I heard. What I heard as well is that if the company wants to go for half dose plus full dose, they need to prove that in a much larger and planned fashion and see that this is not just a chance finding, but that is really something. And I think Peter is right. Is it in which age group does it work? Is it in the younger age group or is it in the older age group? Do we need twice full dose in the older age group? These are all things that we need to find out from the database. And I don't have any of these details, but I'm pretty sure from a regulatory viewpoint, there needs to be much more confirmation.
Any other point, Peter, regarding the way these vaccines are now being developed and presented to the regulatory authorities?
Well, one of the remarks I'd like to make, and I have made a few PowerPoint presentations on that over the last weeks, is how is it possible that we are discussing already today, less than 1 year after the recognition of the outbreak, phase 3 data sets, and we are discussing whether we will have licensed vaccines in a few weeks from now. And I think one of the things is, I think the 3 main things were collaboration worldwide, money that has been put on the table, and regulatory collaboration. To elaborate a little bit on the last one, platform technology acceptance. In March of this year, CEPI and IABS wanted to organize a conference on the acceptability of platform technology. And what does it mean? The nicest example is J&J. They have a vaccine against Ebola, with an adeno 26, and this vaccine is licensed. And for the veterinarian directive in the EU, which has been changed quite recently, on the last page, there is a full page on acceptability of a platform master file once the first vaccine is licensed. However, the regulators have taken us by speech. They have accepted for many files, also for Moderna, for AstraZeneca, for CureVac, and probably I forget some others. They have accepted data from another antigen made by that platform to start with. They didn't skip anything. They have asked to do the tox studies, but they were allowed to do this in parallel with the phase 1. And therefore, we have, on a regulatory platform, gained a lot of time by doing so. I think that is something people should know. There were no corners cut off, but they were done in parallel so that we are now much faster with the data on the table. Another thing is that the regulators, and I'm speaking as an ex-regulator, I'm quite proud on it, that the regulators have done a huge effort in helping people. They lowered the times for scientific advice. Don't forget that in Europe, the national authorities are responsible for clinical trials. Paul Ehrlich Institute, MHRA, the Belgian authorities, the Dutch authorities have given a lot of scientific advice to come quickly to the acceptance of these protocols. I think that is what a message that I would like to give to people that don't know what platform technology means, but it has cut a lot of time in the development. Over.
Good. Thank you, Peter. Maybe I go to Gabrielle now. Gabrielle, could you, yes, you.
You're muting. Always problem to find the unmute button.
You are unmuted. Could you had some comments to give about the whole development of this vaccine and the potential role of COVAX?
Yes, thank you very much for that, Paul. Paul Aubrey. Indeed, as most of you probably are familiar with, together with WHO and Gavi, we have set up back in the spring COVAX, which is actually really a unique and ambitious initiative, I would say. I mean, as we all know, that the demand for COVID-19 vaccines really will vastly outstrip the supply, particularly in the short term, and really to avoid what we have seen for the 2009 H1N1 pandemic, where particularly LMICs reviewed fewer doses much later in the pandemic. We really have come together to try that this is not happening again. So what we are trying is to, well, CEPI is responsible for the developing and the manufacturing of the vaccines, and then also with the help of WHO and Gavi to really fairly allocate those 2 billion doses of COVID-19 by the end of 2021. Initially, these doses will come from CEPI, but in the future, COVAX will also obtain vaccines from additional sources, so not just CEPI. For your information, so there's the COVAX facility, which is a tool that Gavi has developed. It pools financial resources to develop these vaccines, purchase them at scale, and then investing them up and then fund for the manufacturing, so that they are ready when they can distribute it as soon as they are licensed. So there are two ways to participate in COVAX, and that's really depending on the country's income status to really enable equitable access to all countries at the same time, so regardless of income. For the upper middle income countries and the high income countries, there's the pooling of the domestic funds to secure doses for their populations. And by joining COVAX, these countries commit to procure enough doses from the facility and also to make an upfront payment to support vaccine development and manufacturing, which is happening at risk. And then the amount that they are paying is actually a reflection of the number of the doses they want. And then for the less welcome economies, Gavi has established the so-called advanced market commitments, and that's really to support the procurement and the delivery of the vaccines for these countries. And then this will be done by using ODA funds from donors, OECD country donors, to really to incentivize the manufacturers by funding volume guarantees to specific manufacturers for vaccine candidates before they are licensed. So thereby really that they are committing to market-wide demand guarantees and which is available to any manufacturer, and then they can procure and assist in the delivery. So really what I think this tool does is to create economies of scale for a manufacturer. So in the end, to ensure that we all have access to the vaccine at the same time, and not that particularly the LMICs will be last in line, so to say. So as we all know, there are a lot of the high income countries already have started, as Peter already showed, have these bilateral deals with the manufacturers. But what is the nice thing about the COVAX facility is also that it complements these deals, and it allows for allocations of a proportion of the doses in these bilateral deals, that they can also be become part of the facility. And it's really, in the end, it's about in these, I think COVAX has a mechanism in these to guide the countries and the manufacturers, to guide them in these bilateral deals. For example, the bilateral deal that AstraZeneca had with BARDA really it funded not only sort of the availability of the vaccine, but also the clinical studies, the vaccine manufacturing, the technology transfer. So really, therefore, in a way, that deal does not only benefits the US, it benefits also AstraZeneca, Oxford, but also the rest of the world, I think. So I think really I think COVAX can also really have a say in these bilateral deals, so that it's beneficial to the entire world.
Thank you. I think that I would like to go to the other side of the world. Romy, how is it in Argentina today?
Hello, hi everybody. It's fine. Thank you for asking. So I think that I have been listening with a lot of attention everything that you said, and of course there is no doubt that this is a historical moment. In terms of vaccine sciences in general, the rapid response to face this new viral challenge from all aspects, right? From the research aspect, from the regulatory aspect, from the economic aspect, has been huge. I mean, I'm hearing you, and it's unbelievable. I mean, it's interesting to be in this moment where you can see everything that is happening, right? These multiple collaborations between academia, industry, governments, to make it happen, and also to make it happen with equality, right? I hear Gabrielle and and everything that she's saying, and to try to at least think about the rest of the world, and try to at least plan to provide doses for all the world, especially low and middle income countries. I don't have much to add. Just one little thing that I wanted to bring to the table that I think that is interesting also to think about it. You know, we are analyzing, you know, the research and the evidence, the efficacy, safety, correlates of protection, and also from the regulatory perspective that has, you know, brought a lot of challenges that we never imagined, you know, in terms of we need to answer questions that we don't have any answer to. But one thing that it is interesting and I think that requires us our attention is what are we going to do with the community, because you know we are always talking about from you know the research perspective and the evidence and the regulatory and the government and funding and distribution, but I think that we don't must forget that this moment also has brought the community as part of the scientific world as well, because everybody today is a vaccine expert. Every, you know, in every house we have a vaccine expert, right? Everybody talks about vaccines and everybody knows a lot about vaccines and everybody knows about the phases of, you know, vaccine trials. So what we are facing today is the involvement of the community in something that most of them, they might maybe not understand what it really means. A lot of them, yes, but so everybody's talking about that. So my, you know, point here that I wanted to bring up for discussion also, you know, from your perspective is how are going to face vaccine acceptance. We know that we have a lot of very good, hopefully, COVID vaccines, and we know that we are working very hard in trying to get manufactured and to distribute it all around the world. But what is going to happen first with the volunteers of the trials, that we are thousands of people all around the world that have been participating in trials, and second, from the community, if they hear every day news about vaccines and they are talking about they don't know if they trust or not, if they are going to receive it or not. So how we are going to address once we have the vaccine, they have the vaccine in the you know in every country, are we going to manage to do campaigns or to try to make the people trust, because this also will have a very deep impact in vaccine acceptance and vaccine perception and vaccine research as well, because we are now doing a vaccine in 9 months and we always say to the people that we take 10 years. So how we are going to not reveal but you know change and try to transmit this very clear to the communities all around the world. And that's, you know, it's something that I think about it a lot. And I just wanted to, you know, bring to the table, you know, to discuss with you all.
Thank you, Romy. I think that we count on you to be one to convince everyone to be vaccinated. You have been very good
There's
before.
A lot of pressure for me.
But Peter, do you have any comment on that?
No, not really. I mean, this is outside of my area of expertise, but it's obviously a major issue. And what I worry about is that these vaccines are being rolled out initially to people who are essentially frail. And so there's going to be a lot of nasty things happen to these people, completely independent of the vaccine, just because they're elderly or frail. And actually making sure that we have the systems in place to be able to reassure people that, you know, this is what we would expect if you're giving a vaccine to a elderly, frail population, and this is nothing to do with the vaccine. We've really got to be geared up for that. I mean, that we've faced that part of the vaccines. We faced it for HPV vaccines, where we were giving a vaccine to an age group that had really not had much in the way of vaccines before. And we knew things were going to happen in those populations that might be associated by chance temporarily with the vaccine. And this is going to be even more true for these vaccines. So I think this issue about persuading people that these vaccines are safe and they really need to be used is going to be a large challenge.
Yeah. On the other hand, Peter, I'm always struck by the fact that for Shingrix, which is probably one of the most reactogenic vaccine, when Shingrix is given to older people, no one complains. Although, you know, the people receiving Shingrix have many other diseases, many other things they could complain about. They are just trying to get it. And I did not see so far any major issue regarding Shingrix acceptance. So
No,
we may hope that the
mean,
same thing may happen with
COVID
vaccine.
I mean, the major issue with Shingrix, certainly as far as I'm personally concerned, is its availability. I'd certainly have it if it was available here.
Yeah.
But I think my fears are not so much the people who are at very high risk of getting the disease associated with this. I think, you know, take up among the elderly is likely to be high because we know at very much enhanced risk. But it's the sort of younger adults who are going to be crucial in terms of getting herd protected, who will say, well, you know, there may be risks and I'm not really at great risk of this disease, so why should I take the vaccine? And that, I think, is going to be one of the big challenges.
And health personnel, probably. Maybe I go now to Rudzani. You know, we have to go, he's by the Lake of Annecy, apparently. Also a little bit in South Africa, I assume.
Yes, I am in South Africa. Thank you very much. You know, when both Peter and Nick were speaking, I was reminded about the beginning of this pandemic, where someone made a comment that we were all in the same boat. But what became clear very quickly was that we may all be in the same turbulent sea, but others are in cruise ships while others are in log rafters. And I think this is going to become more and more clear. And I'm just going to point at 3 areas, and I'll speak about them very quickly. I mean, Nick spoke about the issue about you know, surveillance and post-licensure surveillance. Right now, we actually, when you look at number of low and middle income countries, and I can speak specifically for Africa, we don't actually know that we are actually capturing the disease accurately as it is. Forget about post-licensure surveillance in terms of disease and the effectiveness. And that's quite key if, for example, it turns out that even the vaccines don't have the same effectiveness in different populations in order to be able to see what's happening in those spaces. I mean, to give an example of South Africa, that actually has better resources than most of our neighbors. The excess deaths are actually 3 times the COVID reported deaths, which means, and I doubt that those extra ones are just due to indirect COVID effects, and will probably COVID cases that we're actually missing because of lack of ability to actually capture and survey the disease. So that's one. The second part, which I think is quite critical, and that has come up when Peter was showing the advanced market pages that is showing. COVAX is like a small blip in the bottom, whereas the more richer countries have basically hoarded all the vaccines already before they were even available for the market. And that's quite important. And for me, what is key is that even the ability and stability and deliverability of that vaccine is going to become an equity and almost a human right issue in this respect. And I think what we'll need to think about, and I'm not sure how we actually do this, if you look, for example, at the 2 front-runner messenger RNA vaccines, they are for all practical purposes not very useful for most poor countries because we just don't have the ability to deliver them effectively to the end user. You know, and I'm not sure if we'll be able to develop capacity for doing so. I mean, I was actually surprised to see the Germans, for example, building these facilities that will allow for one-stop vaccination where you deliver and you vaccinate from there, because moving it from place to place will make it lose the effectiveness. And I think it even affected the UK's prioritization of groups by making sure when it became clear which vaccine will be licensed first, that it changed the prioritization, because the group that would have been initially prioritized will be distant and it will be affected by the efficacy of delivering that. What that means for me is that we probably should be trying to ring fence your kind of vector group of vaccines that are there, like the AstraZeneca and the Gamaleya group, for example, for poor countries in terms of allocation, because those, at least there is enough ability to probably effectively deliver them to the end user in such a way that it allows equity, whereas rich countries can benefit from the other vaccines that are the front runners, that they have the capacity to actually deliver to that population. And I think that's important. And lastly, I think, again, Peter, you referred to it when we were talking about advisory groups. WHO, for example, has issued some advice in terms of how to prioritize groups for vaccination within a country. A number of low and middle income countries only have effective NITEX, especially when things change rapidly. For example, again, giving an example of the UK, the change in prioritization in vaccination could be done very quickly when you realize which vaccine is in the front and that its delivery is actually different from the AstraZeneca one, which they probably had thought would be the first one to come through in the end. WHO has been very helpful, especially for me, the NITEC Support Center, in terms of trying to capacitate poor countries to be able to make contextually relevant and appropriate decisions in terms of vaccination. And again, this is going to be very key, because part of this delivery, which I believe may be based on campaign type distribution, has got huge implications for the other, for the EPI schedules. And these have usually been very disruptive to poor countries. So there are all these things that actually will become very, very clear as the 2021 unfolds, that we really may not be in the same boat. But the question is there anything we can do, in the spirit of justice, fairness, equity, to try reduce the impact that will be carried by mostly poor countries, that and I believe in the end that may also have impact in high income countries. I'll stop here. Thank you.
Thank you. Danny, do you have anything to say about that?
Thank you. So I work in the Department of Immunizations, and I appreciate the comments from Rudzani about WHO's help. I can't take much credit for that, as that's more in the NITIC strengthening group. But I will say that we're also working at WHO on providing guidance for various streams of post-introduction evaluations. There's been a lot of work, probably the most work has been done in the safety realm. And the group working on that has just put out a safety surveillance manual that's available on the web that talks about all the different aspects of monitoring these vaccines for safety. They're also putting together a protocol that countries can use to monitor safety, and have had regular meetings with the regional safety focal points. There's also a group working on programmatic evaluations post-introduction, the so-called PIEs, so that countries can get feedback quickly on how the early introductions went and what lessons can be learned from that to improve vaccine distribution going forward. And then the work stream I've been most involved with is looking at some of the vaccine effectiveness evaluations that Nick was talking about. And we've had a working group that has met for the last couple months. Our concern is that there will be these VE studies will be popping up all over the place. And I think Nick, in some ways, and the UK experience has made it look easy because they have so many well-established platforms that they can leverage. But in many countries, many lower middle income countries, they don't have the platforms. It will be more difficult. And these are not easy studies to do. There's methodologic issues. There's lots of potential bias. And so we want to make sure that they're done correctly. One example that Nick brought up several, but one that he didn't, is an issue of spurious waning, that this question of duration of protection is so important, but it's actually quite difficult to get at, because what tends to happen in vaccines that aren't 100% effective is that over time there will be some protection among the vaccinated group, but it'll be diminishing. Whereas in the unvaccinated group, there's no protection, and when there's high incidence, that unvaccinated group will get more natural immunity quicker than the vaccinated group, and it will make it look like the vaccine effectiveness is going down over time. So there's these issues that need to be considered. And then the last thing I'll say is that we are trying to map out, get some sort of landscape of where these VE studies are taking place globally. What are the target groups being studied? What are the methods being used, so that we can identify where the gaps are in the evidence, what the decision makers, the policy makers need, and hopefully be able to fill those gaps in promoting studies in those areas. Again, particularly, we are concerned that these studies will happen in high income countries, but not in lower middle income countries. Thank you.
Thank you. Maybe I call now I think that Tracy want to speak to the other members of the panel.
Right. So I'm here representing the Caribbean.
Melinda and Sonali. So if you are first there, go ahead.
Yes. So I'm here representing the Caribbean, and just to say that in terms of our information systems, we are putting things in place. For example, the vaccine card that Nick spoke of, we're trying to standardize that for all the Caribbean countries through PAHO Caribbean representative, so that we can be able to have a standardized immunization registry and immunization card. We have our SARI surveillance system is very well established as a source of information. And one weak area is our ISAVI system, which, for the other vaccines, PAHO told us we weren't really picking up much. And one of the concerns were, if we start, improving our ISAVI for COVID, people are going to say, but COVID has all these side effects that we didn't see with other vaccines, but it's really just a program issue. And the other thing that's important is what was mentioned before regarding the vaccine acceptance. And we actually polled the EPI managers, and about 2/3 of them had a concern about vaccine safety and efficacy. So what we're focusing on right now is getting the background feel of what vaccine acceptance and creating communication messages to tailor that. So we're working more on preparing for the vaccines with terms of vaccine programming, so that those things are in place and the acceptance is good. I mean, we're at the bottom of the totem pole in terms of the vaccines that are going to be distributed. And that is actually a good thing, because then everybody else will get it, and we'll get a chance to see how the rest of the world has reacted to it. By the time it gets to us, it might improve our confidence in the vaccine by then. So that's basically what's going on in the Caribbean. We're making preparations to get started, and kind of paving the way so that when it is here, everybody's ready to take it, and the programs are in place and the surveillance is in place. Over. Thank you.
Thank you. Melinda, you have a comment?
Yeah, just a quick word about the situation in the United States. On December 1st, our NITAG, the Advisory Committee on Immunization Practices, met and made a recommendation that should a vaccine be authorized for use in the US, the first groups to be vaccinated would be healthcare workers, broadly defined, including people who were at risk of exposure, even if they didn't have direct patient contact, as well as staff and residents of long-term care facilities, which really would include our groups that have had the highest mortality rates from COVID in the US, and are made up of largely many of them are very frail elderly people. So with that as a backdrop, the FDA's advisory committee is meeting tomorrow to discuss the Pfizer product. Of course, I don't know what the outcome of that will be and whether or not FDA will authorize the product, but if they do, we have scheduled ACIP meetings on Friday and also on Sunday that could make a recommendation for use of the Pfizer product should it be authorized. And if that happens, vaccination could begin next week. In light of the sort of logistical issues of using the Pfizer product, our initial focus, the implementation is likely to be focused initially on large healthcare facilities that will have the infrastructure that they can deliver the large quantities of vaccine that we expect to be in initial orders, as well as long-term care facilities, given the, again, the recommendation from ACIP and the health impact within those facilities. So we are anticipating a program to launch as early as next week.
Thank you. Sonali? You are mute. You are muted.
There are just 2 points that I'd like to briefly cover. One is the utilization of the vaccines in pregnant and in lactating women post-EUA, and the other is the enhanced monitoring program that have been set up for COVID-19 vaccine safety assessment. So in terms of the utilization of the vaccines in pregnant and in lactating women post-EUA, we've seen that there is a significant impact of COVID-19 on pregnant women and neonates. And there's currently absence of data to support a risk-benefit assessment in these populations. Developmental and reproductive toxicity or DART studies are currently ongoing for the vaccines granted EUA or are currently planned. And there are plans or discussions to include pregnant women in the clinical development programs. For example, for the Pfizer vaccine, the DART studies are currently ongoing and should be available soon. And 23 participants had reported pregnancies in the safety database, including 9 participants who withdrew from the study due to the pregnancies. So these participants will continue to be followed up for maternal and neonatal outcomes. And pregnancy registries and post-authorization safety studies or PASS studies, of course, will be set up as part of the risk assessment plan. So data might be available from the utilization of the vaccine platform for other vaccines, and they are standardized benefit risk templates that we have prepared for all the vaccine platforms being used for COVID-19 vaccines to harmonize this data collection. And there's potential for pooling data by vaccine class from follow-up during clinical trials of inadvertent pregnancies, from pregnancy registries and PASS studies. But it's critically important to utilize a standard SOP, a format, and case definitions. For example, the Gaia case definition that we developed for ease of combining data. And lactating women are part of some of the clinical development plans. Information from pharmacokinetic studies of the vaccine might also provide insight. So currently, we do not have the data to support the benefit risk assessment of the vaccine in pregnant and lactating women, but the data should be forthcoming. And in terms of the enhanced monitoring programs that have been set up for COVID-19 vaccine safety assessment, these are to supplement the existing programs. And examples from the US include the V-Safe or Vaccine Safety Assessment for Essential Workers set up by CDC, where essential workers are followed up up to 12 months. And it's anticipated that 20 million plus people will be followed during the first months of the vaccination program. And in the EU, this includes the EMA-funded Access project, which involves 22 research centers in Europe. And there's basically continuous monitoring of the coverage, safety, and effectiveness of the COVID-19 vaccine, and providing background rates of adverse events of special interest. Common protocols are being set up for safety studies. In addition, the Consign project will collect data on the impact of COVID-19 in pregnancy to guide decision-making about the vaccination policies and vaccine indications in pregnant women. So a coordinated and collaborative approach is going to be critical for successful COVID-19 vaccine safety monitoring. We already have harmonized protocols, templates, and tools available which should be utilized globally. And real-time global data exchange is going to be important, as pooling of AEFI reports into larger databases will help identify potential safety signals. And collaboration between stakeholders is key, including the national regulatory authorities, public health authorities, vaccine manufacturers, and international organizations. And a lot of these points were included in the WHO Safety Surveillance Manual that we've helped prepare. There's a lot of work that's already been done to take this forward. Thank you.
Thank you. And in India, Manoj, you have any comment on that? How is the situation there?
Yeah, thank you, Paul and Philip. India is always leading on the tally of the number of cases, and this very soon probably will reach to the number one. But the situation, as we have been hearing also the other panelists, that we are simultaneously preparing for the vaccine introduction along with the process of fighting with the disease. Although the vaccine which are to be inducted have not been finalized, whether they are going to use single vaccine or multiple vaccines. And you know about several candidate vaccines which are in the phase 3, although the results of the 3 vaccines are out, but only one of them is starting the clinical trial in India now. So it is still yet to be, I think the data is yet to be identified. Although some of the manufacturers have submitted the data for emergency authorization now. But from the data which is available from the different vaccine trials, the use is something like the disease profile and the background rates are not yet clear in some of the developing countries, at least, because the outcome, the disease mortality and all the profiles are not yet clear. So these will have challenge while we are preparing for the AEFI and the causality assessment things, and assigning a vaccine associatedness or is a signal or is unrelated thing. The other issue would be when multiple vaccines do become available, whether we will use them interchangeably, or how do the vaccine management and administration will be handled. In some countries, there may be only a single vaccine, but I'm very sure that India will actually adopt multiple vaccines, and we are not very sure how to handle that situation. Surveillance in India for the AEFI is relatively passive till now. Now, how it evolves to an active surveillance of the vaccine is yet to be seen now. And when we do not have the background rates and we now move to the vaccine safety surveillance after the vaccination, which may start maybe in a couple of weeks or so, what the anticipations are. So there will be some challenge in the initial safety signals and capturing the cases or so. One more thing which is there, currently the vaccine safety surveillance has not identified any timeline for reporting the adverse events related to the COVID vaccine. So how long the safety surveillance should continue for each of the individual, and which are the conditions, although they have been identified, the AESIs and the AEFIs, but how long somebody should keep on tracking or monitoring, or should mentioning that, okay, I have been vaccinated with the COVID vaccine. The challenges with the efficacy and also the adverse events across the different population from the different countries. Till now, I think I have not come across any data which is saying that the adverse events are different across different populations or different countries or so. But the disease profile is definitely different across the different countries and the severity. So whether the adverse events are also going to differ across the population as the age groups, that has to be seen. The trials probably have not addressed that whether the prior exposure has affected the effect of the protection and also the subsequent adverse events. So that has to be seen now, because here there may not be any discrimination based on the prior antibody level or something or not. So we have to see some of these unanswered questions through the newer, the times to come. And we are chartering, as somebody was saying from South Africa, that, okay, we are chartering on some on the vessels, some on the stars groups, but some of them are also in the same country are traveling in different vessels or different ferries. So only a strategically phased introduction will also trigger for some vaccine eagerness, the desire, the confusion about the prioritization, the hesitancy, unclarity about prior exposure and antibody level, and now need for the vaccine. Some of these are going to emerge, and we might see a new phase in the vaccination era. And that's all. I think we need to keep a track on them and keep on thinking. Thank you.
Thank you. Now I go back to Philip. I think, Philip, we still have some questions which have been unanswered, and you are the one monitoring that.
Thank you very much.
Take over.
Okay, thank you very much, Paul-Henri. And indeed, and despite the fact that Nick Andrews has been doing quite a nice job in answering directly in writing to specific questions, actually quite a few. And the fact that many of the comments that were made by panelists or some of the later discussions, as in my view, address some of the initial questions. Let me try to, because of the very limited time, to put some of what I see as additional key points or things that repeatedly came through the questions or comment. The first thing is, sorry, in no particular order, but the one I put them on a piece of paper. I think there was serious concern about breastfeeding. So coming back to what Sonali said, the concern was more about the breastfeeding and what it may do to the vaccine acceptance or to the breastfeeding itself, because of many healthcare workers being frontline are also potentially breastfeeding in many countries, and with the lack of data and the worry for live or vector vaccine. Another question that also was not addressed has to do with do we have any information on sex disaggregation of the information from the trials, or is there any plan for sex disaggregation of data? Then there is the question about, or rather than question, but it's both question and comments about mixed vaccine, and it came several times in the discussion, the fact that perhaps one should envision a prime boost with different products. The access to vaccine, I think that despite the talk by Gabrielle about the COVAX facility and also the excess doses that can be ordered by some countries, then that would be donations, hopefully. There's still some concern by LMICs about how they could go and secure additional vaccine. Then coming back to the point that Manoj just made, and I was happy that actually surfaced, the issue of those that have been previously exposed to the disease, and that would have done either almost inapparent or inapparent disease, but would have been positive in PCR, or those that would have done heavy duty disease and would have a high antigen, then that's a serious issue to be able to disentangle in the follow-up. And then the question is, how can we really document in the context of the post-market trial a previous exposure? And then some of the participants are actually also concerned about the safety issue for people that may opt themselves to leave the trial, that then rush to get additional vaccines and may end up with receiving 3-4 doses, God knows what, in some setting. And there's one also, there was a debate about the comment made by Stanley about the vaccination of children and mandatory vaccination that was supported by some, but some other participants were not so supportive, thinking it's a misrouting potentially of the limited doses to think about the herd immunity indirect effect. And then some of the participants are also thinking longer term and also forecasting the next pandemic and saying that we realize now that there are some challenges with the trials, the continuation, and these had not necessarily been anticipated before. And they kind of say that we should already be thinking or not lose sight of the learnings and get that addressed early on for the next pandemic to come. Paul-Henri, I think I try to do my best not to collegially address some of that. Some were comments, some are calling for an answer, if any, like the sex disaggregation. So I will turn back to you to address it to any of the speakers or panelists before we close, because we are nearing the end time.
Yeah. Okay, I open it. Who would like to enter, Peter?
I can just take a couple of those. The sex disaggregation, I think that is going to be important, but I mean, we do have data on that so far from the Pfizer BioNTech vaccine, which in the data they've submitted to FDA, and essentially the vaccine seems to be sort of 96% protective in both males and females. So that so far, there's, at least for that vaccine, we see no difference with respect to gender. And then the other question about giving the vaccine to previously exposed, most of the trials have actually included previously exposed in their trial population, and some of their analyses are focused on that total population and some just on those who've got no evidence of previous exposure. But interestingly, again with the Pfizer vaccine, because they've got both groups there are a number of cases in people who, in their short follow-up, who actually had disease in the control arm, who had evidence of previous disease as well. So that, and that to me, a rather surprising number actually. And so that and not so many in the vaccine arm. So it did look, albeit based on very small numbers, that it actually was beneficial to people who'd had a previous infection to get the vaccine again, because we know from other studies that those people are not at zero risk. And there was, albeit not overwhelming, but suggestive evidence in the Pfizer trial that the vaccine will be beneficial, at least in the short term, to those who have had a previous infection. Over.
Good. Thank you. Yes, Sonali, you are muted.
Just to answer the other questions about the additional vaccines being given. So there are some discussions in the EU countries about the additional vaccines being given to COVAX. It's the liability concern that's going to come up because the governments essentially for some of these vaccines which are being offered at not-for-profit, like the AstraZeneca vaccine, then the country governments would have to pick up the liability for the vaccines. So that's the liability concern is coming up in the discussions of the EU, excess vaccine being given to COVAX, but that's being currently discussed. And in terms of placebo recipients receiving 3 to 4 doses of vaccines, we're currently working on a WHO ethics guidance on placebo, placebo groups being continued in the trials, and that guidance should come up, and this would be addressed in that. Over.
Thank you. Other comments? Yeah, Melinda, you are muted.
Yeah. This issue of trial continuation is on the agenda for the FDA Advisory Committee meeting tomorrow. I don't know what the speaker is going to say, but it is part of that discussion. So I'll look forward to hearing how that goes tomorrow at FDA.
Good. And also with all this variety of vaccine being developed, as mentioned by Manoj, we can expect that people will mix one vaccine with another one for the second dose, and it's maybe a mess too that we have to try to avoid.
Yeah, I mean, certainly, I think in the UK, our sort of vaccine czar, I don't think that's her name, but something like that, has actually discussed this as a specific issue and has raised the possibility of actually doing trials where they get the initial vaccine with one vaccine and then boost with another to see if in fact it may be more effective. So certainly doing that in a controlled way would be much better than doing it in an uncontrolled way, but clearly both are probably going to happen. Over.
Yeah, Well, it has been done in the past for hip vaccine.
I think something we haven't looked at are the dropouts, the people who only take the first dose and don't complete the course. It's going to impact on how we, interpret the efficacy with that cohort of persons.
Okay.
Philip. And I as an activist will probably have to mention that I think in the long term, how we find a sustainable model for these going forward will become important, because the assumptions in terms of currently in terms of resources being looked for poor countries don't assume that we're going to be needing, as Stanley pointed out, vaccine for this in perpetuity. It's almost like we've got this and everyone will get their regimen, whether it's 2 doses, and that's it. That's basically how we're thinking about this. But if we end up with a disease that is continuous without longevity of protection, we're going to have to find a sustainable model for this. And given that it probably looks worse than flu, and we don't even have sufficient for that, it's something to just worry about what a sustainable model could look like for ongoing vaccination.
Philip, back to you.
Okay, thank you very much. I would like to thank all the participants for their contribution, for having been part of the webinar and contributing. A special thanks to our panelists for your truly excellent contributions. I mean, it really covered a lot of ground and issues. Special thanks, of course, to our outstanding presenters, Peter and Nick, and our wonderful chair. What I didn't say about Paul-Henri, he left because he thought he was aging, so he left the directorship of ADVAC. But the more I see him, the more I think he is actually getting younger by the year.
You know that after escaping ADVAC.
You should come back, but I know you have not left ADVAC. But in any case, thank you so much, Paul-Henri, for your continuous support. I want to thank Carol, who is doing a lot of work for the organization in the background. So thank you, Carol. We cannot see her. We need to equip her with a state-of-the-art computer with a video camera. And I want just to conclude wishing all of you a happy holiday season and a successful deployment of COVID vaccines. So thank you, and hope to hear good news in a few weeks. So thanks again.
Thank you. Bye-bye.
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 Thank you.
Thank
Bye-bye.
you. Thank you.
Bye, everybody. Bye.